The overall goal of the following procedure is to assess the effects of a range of wavelengths and intensities of red, near-infrared light therapy on oxidative stress in vitro. In order to do this, we have developed a novel method of delivering the light where we can manipulate the wavelength and intensity delivered. We mount a xenon or broadband light source in a housing on a metal base, then place a collimating lens, water filter and entrance aperture in front of the light. The next step of the procedure is to guide the light generated from the broadband light source through the entrance aperture and liquid light guide and through a second collimating lens. Then, bandwidth and neutral density filters are fitted to obtain the desired wavelength and intensity of the light. Note that the filters allow passage of a narrow range of wavelengths referred to as a waveband. The main advantage of this technique over existing methods of red near-infrared light therapy delivery, such as LEDs or lasers, is that it allows precise and calibrated manipulation over wavelength and intensity parameters. This then allows us to optimize the delivery of the light for the best biological outcomes. The current study administers light to disassociated cells. In addition, it may also be applied to other systems, such as organotypic models of neurotrauma. When working with any bright, broadband light source, ensure that you do not look directly into the source of light, as it may result in permanent visual damage. To prepare the light delivery apparatus, connect a broadband light source, such as a xenon or tungsten lamp, to an appropriate power supply. Position a collimating lens in front of the light source to produce a collimated beam of light.